Dear students, we are going to start a new chapter, instrumental methods. So in this method, we are going to study different instrumental instruments uh, which are used in analysis of molecules. So the first point is a relation between the analyte stimulus and the measurement of change in the absorbable property. Now see here, there is an relation between a compound, the stimulus, that is if compound absorbs light, and then after that the third part is the result. Now see here, analytical chemistry deals with the identification, characterization and estimation of the components of sample. So in this branch, we are going to identify a compound, we can characterize that compound, and then estimation of that component of a sample. The component of interest in the sample is called as analyte. Suppose a nicotine is there, from the leaf of that plant we extract exactly the nicotine, so that interesting part or interested component is nicotine of that sample. That nicotine is known as an analyte. Earlier the components of interest in a sample were separated by precipitation, extraction or distillation. So earlierly, for the separation of these interested components of that sample are carried by precipitation, extraction or distillation. At present, highly efficient and sensitive chromatographic and electrophoretic techniques are used for the separation of the components of complex mixture. So nowadays we are using different instruments for the separation and identification of these interested components from the sample. Qualitative analysis gives information about what is present in the component. Qualitative analysis gives information about what is present in the component, and qualitative analysis indicates how much is present, that is the amount of component in the mixture. So you must familiar with these two words. One is qualitative, another one is quantitative. So qualitative means what is present, and quantity means how much is present in the sample. An instrument for chemical analysis converts information about the physical and chemical properties of the analyte into information which can be easily interpreted. So what instrument do? Instrument converts the physical or chemical properties of that component into the information which is received in terms of values, numbers or a graph. Thus an analytical instrument is a communication device between, now see here, the analytical instrument is a communication device between the component, that is analyte, and the investigator. Suppose we are doing an experiment in a lab, then we are the investigators, and the component which we are going to investigate, that component is an analyte. So that instrument is a middle that gives information about that component to the investigator. So it is a communication device. To get the desired information about the analyte, a stimulus has to be provided. Now see here, if you want something information from that component, if suppose the structure is there, the angle is there, the rotation is there, so such physical or chemical properties of that component are collected by use of a stimulus. So a stimulus has to be provided, which is usually in the form of energy. So we may provide energy, electromagnetic, electrical or mechanical or nuclear. So this stimulus is given to the component in terms of this electromagnetic radiation. We may incident the molecule or component by use of IR radiations or different UV microwave radiations. Secondly, we can stimulus by use of electrical or by use of mechanical or by use of nuclear. So the energy is provided to that molecule in terms of these different stimuli. The stimulus elicit a response from the analyte which can be measured or interpreted. If suppose a structure is cubic is there, a different structure is there, so depending upon the structure, that molecule gives response to that respective stimulus. For example, they have given here example: a visible light is passed through a colored analyte solution and the extent of its absorption by the analyte is measured. Suppose a colored solution is there. Now see this block diagram here. This is the colored solution and we are passing a source of light, suppose, and some amount of light is absorbed by that colored molecule or colored solution. So how much is absorbed is given by the instrument. The amount of light absorbed is correlated with the concentration of the analyte. If more the concentration of that component in that solution, then absorption is more, because light cannot be passed completely through that solution. Therefore, the amount of component is calculated. Now we are going to study the block diagram of an analytical instrument. An analytical instrument measures the response of the analyte to the stimulus and correlates it with the analyte concentration. So what instrument do? The response of analyte to the stimulus, it is related with the concentration of the component. Now see this diagram. Here on the left side, source of energy is there. On the right side, analytical instrument is there, and middle part is an analyte under study. So whatever we are going to study, that component has to put into the instrument, and the source of energy in terms of electromagnetic radiation, or in terms of electrical, mechanical, or in terms of nuclear. So we have to provide that energy to that solution, and then we are getting the response in terms through the instrument. So electromagnetic radiation is used as a stimulus in many instrumental methods of analysis. The analytical instruments measure the response of the analyte to the radiation. The response should be proportional to the concentration of the analyte. So whatever the response we are getting, it is proportional to the how much concentration of that that component in the sample. So there are some examples. Now see the first example here, a spectrophotometry. In that spectrophotometry, absorption of UV or visible or IR light is measured as a function of concentration of that analyte. So how much light of UV or visible or IR light is absorbed by that component is correlated with the concentration of that component. The second is turbidimetry. Scattering of light is measured as a function of analyte concentration. If light is how much light is scattered from that component of that solution is given by the turbidimetry. Third, in refractometry, refraction of light by the analyte solution is measured as a function of its concentration. In the fourth, that is polarimetry, optical rotation of PPL, that is plane polarized light, is measured as a function of the concentration of the analyte. In fluorimetry and phosphorimetry, emission of light is measured and correlated with the concentration of analyte. You have seen here, there are 5 examples they have given: one is spectrophotometry, turbidimetry, refractometry, polarimetry, and fluorimetry and phosphorimetry. So in spectrophotometry, how much amount of light is absorbed is correlated with the concentration. In turbidimetry, scattering of light phenomena is correlated with the concentration. In refractometry, refraction of light is correlated here. In polarimetry, how much rotation of that plane polarized light takes place in the instrument is measured and is correlated with the concentration. In the last instrument, that is fluorimetry and phosphorimetry, emission of light is measured and it is correlated with the analyte. So every instrument has its own function. So spectra has absorption, turbidimetry has scattering, refractometry has refraction, polarimetry has the rotation of light, in fluorimetry the emission of light is there. So these are the some examples through which we are studying, or which we are going to calculate the concentration of the analyte from the solution. So the next point is types of analytical instruments. Uh, Analytical instruments are classified depending on the optical, electrochemical, and thermal properties of the analyte which are going to be studied. So, depending upon the optical, electrochemical, and thermal properties of that analyte, that is analytical chemist has differentiated these instruments in different parts. The first one is an optical properties. If you are going to study the optical properties of that component, then the instrument is different. Now we are going to study the first is optical properties. Now see here, optical methods of analysis are based on the interaction of analyte with the radiation. So in this case, in optical methods, there is an interaction of radiation and the analyte, that is component. The components of a sample interact with radiation in different ways. Suppose some component absorbs the radiation, or they emit the radiation, or there is a scattering of light takes place. So depending upon the interaction of that light with that component, again these instruments are again differentiated. The intensity of light absorbed, emitted, or scattered depends on the amount of analyte in the sample. How much analyte is present in the sample, depending upon that concentration, the light may absorb, may emit, or may scattered. So the first instrument is a spectrophotometry. Now see here what happened in this spectrophotometry. In a photoelectric colorimetry, I think you have studied in the last FY class. In a photoelectric colorimetry, the amount of visible light absorbed depends on the concentration of light absorbing components in the sample. How much light absorbing components are present in the sample is directly proportional to the amount of visible light absorbed. If more the concentration of light absorbing component in the sample, then the amount of light is get absorbed more. UV absorption is mostly used for identification of components. So UV absorption mostly used for the identification of components. IR radiation is used for the structural elucidation of the component, that is mostly the functional group identification. If suppose there is an carbonyl functional group is there, or different functional groups, ether, alcohol functional groups are there, so these functional groups are specifically identified by use of IR spectrophotometry or by use of IR radiation. In turbidimetry, the amount of light scattered by the analyte is used to determine its concentration in solution. How much concentration of analyte is present in the solution is directly proportional to the amount of light scattered. So this is a spectrophotometry first. And the second, that is polarimetry. Now see here what is exactly the phenomena takes place in this polarimetry. Substances with asymmetric carbon atoms. Now see here, those substances which have asymmetric carbon atoms. Asymmetric carbon means those molecules in which the carbon is bonded with 4 different atoms. Suppose a central carbon is there, it is bonded with A, it is bonded with B, it is bonded with C, it is bonded with D. So if A, B, C and D, so all these bonding atoms to the central carbon is different, then they are known as an asymmetric carbon atom. So such substances which are asymmetric, they rotate the plane of polarization of polarized light. They rotate the plane of polarized light. This property of rotation of light, now see here, this property of rotation of light is used in polarimetry to determine the concentration of the optically active compounds in the solution. So how much concentration of the component, that is optically active component, is present in the solution is determined by use of this phenomena. So in this polarimetry, those molecules which are optically active, they rotate the plane polarized light or they rotate the plane of that polarized light to the right or left. So depending upon the rotation, the components can be identified. For example, sucrose rotates the plane of polarization of polarized light to the right, so all sucrose are dextrorotatory. Glucose is also a dextrorotatory, but fructose rotates the plane polarization to the left. So glucose is a dextrorotatory, means the plane of polarized light is rotated towards the right side. In fructose, if the light is incident on that compound, that is fructose, the plane of that light is get rotated towards the left side, that is leo-rotatory. So depending upon the rotation, depending upon the amount of rotation, the components can be identified by use of this polarimetry. So these first two are, in this case, these are the optical properties. The second is electrochemical properties. Now see here, electrochemical methods of analysis are based on the measurement of electrical properties of the sample. So in this case, we are going to measure the electrical properties of the sample. So electrical properties of the sample are measured in these electrochemical instruments. The methods are subdivided depending upon the electric property which is being measured. Electric property which is being measured and the conditions under which it is measured, means which property, which electrical property is going to be measured. Depending upon that, again these electrochemical methods are differentiated into a potentiometer, conductometer, voltammeter. So again voltammeter are differentiated into polarography and amperometry. Colorimetry is there. So the first we are going to see the potentiometry. So in electrochemical properties, in electrochemical methods, the electrical properties of the sample is measured. The first, now see here in potentiometer, in potentiometric measurements, the sample solution forms part of a galvanic cell. So sample solution forms a part of galvanic cell. The potential developed in the galvanic cell is related to the concentration of the sample solution. So in this potentiometer, the instrument, the potential which is developed in the galvanic cell is correlated with the concentration of the sample solution. In potentiometric titration, the EMF of the electrochemical cell, now see here, EMF of the electrochemical cell setup is measured after the addition of successive installment of the titrant. The endpoint is determined from the graph of EMF versus volume of titrant added. So what we do in potentiometer, so we set the potentiometer, we fill the titrant in B red, and the component which is going to be measured is taken in the a beaker, and a potentiometer cell is dipped in that beaker, and we successively add a titrant from the B red into the conical flask at different time interval, and we titrate that solution from the B red to into the conical flask, and how much potential is differentiated, that potential is noted on the paper. So by use of that points, or by use of that numbers, we plot the graph of EMF versus volume of titrant added, how much volume of titrant added and what EMF is changed, how much EMF is changed. So that graph gives us the concentration of the component in the solution. So example is an acid base titration. The second instrument is conductometry. The conductance of the sample solution is measured and related to the concentration of the sample solution. Means in that conductometry we calculate, we measure the conductance of the sample. In conductometric titration, The conductance of the solution is measured after the addition of successive installment of the titrant. So the same case with the same case which is discussed in potentiometer, uh, the same titration is carried in conductometry. Only difference in between the potentiometer and conductometer is that in potentiometer we measure the potential developed in terms of EMF, and conductometry we measure the conductance of the sample in a titration. The example is same acid-base titration. The third instrument is an voltammetry. The sample under study is made a part of electrocyclic cell, and the current passing through the cell is measured at different potentials. So in this voltammeter, the sample which is going to be studied is made a part of electrocyclic cell, and current passing through the cell is measured at different potentials. The current measured is related to the concentration of electroactive species present in that solution. So I think you have studied this voltammetry in 12th standard. The next is in polarography, that is the part of voltammetry is a polarography. In this voltammetric technique, now see here, a substance is identified by measuring the electrical potential at which it is electrolytically deposited from its solution. Means what happened here, a substance is identified by measuring the electrical potential. Substance is identified by what we are going to measure here, by measuring the electrical potential at which it is electrolytically deposited from its solution. The concentration of the solution can be determined by measuring the current involved in the deposition process. So at what current the component is deposited is calculated and it is correlated with the concentration of the solution. So a dropping mercury electrode, DME, dropping mercury electrode is used as a cathode on which the substance gets deposited. For example, so this technique is used in the estimation of metals present in the solution. So how much metal is present in the solution is measured in terms of polarographic, that is dropping mercury electrode technique. So in what now see in second, amperometry. In this voltammetry method of analysis, the potential applied is kept constant. The current passing through the electrolytic cell is measured after the addition of successive installment of the titrant. That is, the current passing through the electrolytic cell is measured after the addition of successive installment of the titrant. The endpoint is determined from the graph of current versus volume of titrant added. So in this case, we measure the endpoint of that experiment by use of a graph where the current is plotted against the volume of titrant added during the this experiment. The amperometric titrations are used for the estimation of several metallic analytes which can be deposited on a mercury drop cathode. So we estimate different metallic analytes. They have given the example here: bismuth rice plus versus EDTA, magnesium twice plus. It can be titrated by use of 8-hydroxyhydroquinoline. Lead by use of K2Cr2O7, potassium dichromate. So these two, polarographic method and amperometry method, these two are the voltammetry method. In voltammetry method, the sample which is under study is made a part of electrolytic cell, and the current passing through the cell is measured at different potentials. So the current measured is related to the concentration of the electroactive species which is present in the solution. So next is in the fourth point of this electrochemical properties is colorimetry. In this electrochemical technique of analysis, the sample solution is made part of an electrolytic cell and electrolysis is carried out. The quantity of electricity passed through the solution is correlated with the concentration of electro-oxidized or electro-reduced species in solution. example is an electrogravimetry. So what actually we do here, in this technique, the sample solution is made a part of an electrolytic cell and electrolysis is carried out. And the quantity of electricity which is passed through the solution, it is correlated with the concentration of electro-oxidized and electro-reduced species which is present in the solution. So we have seen here, one is optical properties. In optical properties we have studied spectrophotometry and polarimetry. In spectrophotometry, absorption of light is important. In polarimetry, a rotation of light is important. In electrochemical properties, we have seen a potentiometry. We measure EMF, that is potential difference, which is developed in the cell. In conductometry, conductance of the solution is measured. In voltammetry, that is the current passing through the solution is measured at different potentials. In polarography and amperometry are the part of voltammetry. Coulometry, in this case, the same here, the sample solution is made a part of an electrolytic cell and electrolysis is carried out here. The third property by use of which we differentiate again the instruments is a thermal property. In thermal analysis, now see here, some physical or chemical property of the sample, is measured as a function of temperature. So in this case, in this analysis, we measure physical or chemical property of the sample as a function of temperature. When a matter is heated, it undergoes certain physical or chemical changes. So when we heat the compound, the physical or chemical properties of that component is get changed, or both over a wide range of temperatures. So if we start heating the component from lower temperature to higher temperature, the physical or the chemical properties of that component goes on changing. So that property is used for the identification or identification of the component. Physical changes like melting and chemical changes like dehydration or decomposition of the sample are studied over a range of temperatures. So in this case, melting is a physical property and the dehydration or decomposition of the sample is a chemical property, are studied during this experiment. These changes are characteristics of the sample under study. So these changes, the change in melting point or change in the decomposition of the component, are the characteristic of the sample which is under study. By determining the temperature at which these changes occur, it is possible to characterize the components present in the sample and thus identify them. So determining the temperature at which these changes occurs, so at which temperature the melting point change, that is the melting point takes place, the melting point of component takes place, so that temperature gives information about the component present in the solution. So the thermal methods are again classified into TGA, DTA, DSC and TT. So the first one is thermal gravimetric analysis or thermogravimetry. Now see here, thermogravimetry. In thermogravimetry, the mass of the sample is measured as a function of temperature. That is, mass of the sample is measured as a function of temperature. That is, the change in weight of the sample is measured at different temperature. So at different temperature, we measure weight of the component. So that mass of sample is measured as a function of temperature here in TGA. The second is different thermal analysis, that is DTA. So in this case, in which the difference in temperature between the sample and a reference material is measured as a function of temperature. That is the difference in temperature between the sample and the reference material. So that difference is measured as a function of temperature. The temperature difference is equal to Ts minus Tr. Ts means the sample and TR means the reference material. So the difference is measured, and that difference gives us the information about the component which is present in the sample. Third one is differential scanning colorimetry. So in which the energy required to establish a zero temperature difference. Now see here, the energy required to establish a zero temperature difference between the sample and reference material is measured as a function of temperature. So how much energy is required to establish a zero temperature difference between the sample and reference material? So that property is studied in the differential scanning colorimetry. And last one is thermometric titration. In thermometric titration, the changes in temperature are measured during the course of titration. So changes in temperature during the course of titration are measured as a function of amount of component present in that solution. So example is acid-base titration and complex matrix titration. So these are the types of instruments depending upon the properties they have differentiated here. So just in 1 minute I will revise this point. So what we are going to study here in this topic is different instruments and use of these different instruments for the analysis of the component. So we must know what is meant by qualitative analysis and what is meant by quantitative analysis. The block diagram of an analytical instrument. In that block diagram, the energy is passed through the solution. This energy may in terms of an electromagnetic radiations, or may in terms of electrical energy, or may in terms of mechanical or nuclear energy. So this energy is given to the component, and this component gives response to that energy, and we are getting a result through the instruments. So there are different instruments: spectrophotometry, turbidimetry, refractometry, polarimetry and fluorimetry. We have studied this and the properties of that instruments. So there are different types of analytical instruments. By use of optical properties, they have differentiated spectrophotometry and polarimetry. In electrochemical properties, potentiometer is there, conductometer is there, voltammeter is there, and colorimeter is there. And last is the thermal property. By use of thermal property, again the instruments are differentiated into TGA, DTA, DSC and TT. So in the next point, we will start spectrophotometry.